Four biomolecules. What are your four molecules? Nucleic acid, carbohydrate, lipid, and protein, right? They told you them. What are their monomers? What's the monomer of nucleic acid? Nucleotide. What's the monomer of carb? Monosaccharide. Here, I'll zoom in a little bit. What is the monomer of a lipid? Fatty acids. Don't forget about glycerol. What's the monomer of a protein? Amino acids. Good. Sticking. Good. Question 2 asks you what do you look like? What's your functions? What's the function of a nucleic acid? Genetic. Little protein synthesis. What about a carb? What a carb did? You probably eat these for breakfast. Why? Energy. And a lipid gives you long-term energy. They're also a big part of that cell membrane. Okay. What about a protein? I gotta work on my protein goals. Why? Why I gotta work on my protein goals? Proteins gives me. Gonna form cell structures. It'll be enzymes, cell transport, immune system, hormones are all proteins. It's a little bit of everything. That's why we say cell structures. It does a little bit of everything. Good. That was biomolecules review. Page 78 was your enzyme review. What's the role of the active site? What do active sites do? There where I. What is the role of the active site? It's the binding site. It's where it attaches. It's where it's going to do our little divide, where it's going to either break stuff down or help it form stuff together. Is it specific or not specific? Specific. Remember, it is a lock and key. Lock and key. What type of biomolecules are enzymes? Proteins. What effect do enzymes have? What do they do? What's the purpose? Speed up reactions. What's an example of an enzyme? Lactase. Right? You can take a pill of lactate. It has lactase in it. It's going to help you break down milk, lactose. Lactase means enzyme. So what does it mean to be denatured? I broke it. It's not going to work anymore. What are the two things that will break down that enzyme, cause it to become denatured? What are the two things that are going to cause it to become denatured? Temperature or pH. Good. Temper pH is what's going to break it. Good. All right, for today, flip to page 20. Oh, you're not ready. Flip to page 29. 29. Thanks. 29. Your topic for today is all about scientific method and enzymes. You're going to spend all your stations today on enzymes again. So your recap sheet is just reviewing the scientific method, making sure that you understand what's going on, making sure you can interpret graphics and associated with enzymes. Sorry, I didn't mean to throw it at you. So our scientific method, what do we start with? Always. I'm about to become a scientist. What do I do first? Make observations. I'm going to observe some phenomenon. I'm going to look at something, and then I'm going to ask questions about it. I'm going to say, hey, I can observe that there are about 20 people in this room. I wonder what everyone had for breakfast. Does breakfast affect how you do in class? I start asking questions. When I formulate a question, a big one that pops up is what we call a testable question. These are ones that we can run experiments on. They're typically based on data. If I ran an experiment and I said, I wonder what was everyone's favorite breakfast, would that be a testable question? Could I test that? No, right? Because you might have a favorite breakfast. You're like, I love pancakes. But if you give me French toast, I'm going to pick French toast. Right? It's an opinion. I can't run an experiment on that. You might change what your favorite breakfast is based on what you tried. I can only do it on things that are going to be concrete, things that I can repeat. Remember, I have to be able to repeat it 3 times. Someone else got to be able to do it. I can say, does breakfast increase test scores? I can see that regardless of what you ate. I give you a standardized test, people who ate breakfast, people who didn't. I can run a test the question. After that, I formulate what we call a hypothesis. What is a hypothesis? You should know that one. What is that? It is a educated guess. Based on observation. How do I write it? I always write it in a format. If I do blank, then blank will happen. If I do blank, then blank will happen because blank. This is where you introduce that science knowledge. This is what makes it educated. If I eat breakfast, then my test scores will go higher because carbs give you energy and you need energy to use your brain, right? I'm going to give it more info and I'm making my guess based on something I know. Typically, my X and Y are going to be my variables, things I'm going to change and measure. Your IV is what you change. That is always our X variable. DV is what I measure. Your Y variable. What would be constants then? What I keep the same? So in terms of my breakfast and test scores, maybe everyone takes the same test. We're all in the same room. We all get the same amount of questions. We all get the same time. What's a control group? The one that doesn't get the IV, right? This is my baseline. This is my comparison. It should be considered your normal group. So I need this to compare to. So in the case of breakfast increasing test scores, my control group would be the person who doesn't have any breakfast. Last part of this page is your graphs, your different types of graph. In bio, you mostly are going to use a histogram, a bar graph, a line graph, or a pie chart. Graphs are very good at giving you your information. It's going to give you your IV and your DV. Your x-axis is always your IV. Your y-axis is always your DV. The graph typically will give you a title telling you what you're working with. It's going to have labels. Those labels are going to have units on it. It's going to give you a bunch of information just by the labeling of the graph. You are not going to be required to make any graphs on your SOL, but you are going to be required to interpret them. So a big thing is to note, what is the label saying? What is the IV? What is the DV? Can you pull that information from a graph? So histograms are always for variability, something about frequency, how many times does it occur. Bar graphs are always for comparison. Line graphs are either for correlation, they're always numerical, and pie charts are typically percent of a whole. And that is it for today. Yes. Yes. What you're going to do today, you're going to go back through enzymes. You're going to need to reference your pages from last stuff, talking about enzymes. You're going to reference back to page 8 if you don't understand what you're doing. You have 3 stations to do, stations 1, 2, and 3, about your enzymes. You are missing words in task 3. You're going to add the suffix ASE and lock and key. You are missing words. A lot of people were confused about that. Station 2 is going to be multiple choice. Station 3 is where you start getting your graphs. Can you interpret the graphs? Your quiz on page 34, you're going to the bottom of 34, and then you will turn your book in. Does anyone have any questions? Okay.